Good morning, ladies and gentlemen. I'm going to share some of the research work we have just started in Flinders University in the School of Biological and Chemical Sciences. What we are looking at, we want to assess the future impact of seawater desalination plants on the marine environment. As we know that the freshwater scarcity will increase with time. So how we'll tackle this problem? If you look at a desalination plant, it looks like a typical industrial complex. However, it's emerging as a promising technology for efficient production of fresh water from seawater. And desalination plant at Stattenweg in Adelaide is expected to produce almost 50 gallon liter of fresh water every year, which will meet about 25% need of Adelaide water supply. And this will go up to 100 gigaliter of water in time. What we see here, something is missing, okay, is that desalination process is already utilized in a lot of countries for the production of water. And at that moment, there are about 15,000 desalination plant operative in 155 countries. Out of them, almost half of the capacity is in Middle East, and it's obvious because in Middle East there is a scarcity of freshwater sources, and second, they have access to very cheap energy. Saudi Arabia is the biggest producer of fresh water from desalination processes, and second to this is United States of America, which produce about 12% of the total water produced all over the world. And in time, we expect that number of these desalination plants are increasing, and there will be many all over the world in coming decades. There would be obvious associated problems with it because there are good reasons why we need more fresh water. There is acceleration of world population growth, there is a increased consumption of natural resources, and third, there is climate change. And all these three will combine to have a dramatic declining effect on the access of fresh water in the world. So how does this desalination process works? We can break this into major three components. There is an intake of saline water from sea to feed the plant, and then plant will pre-treat this water, and that under pressure it will pass through a membrane which is a semi-permeable membrane, and at the end of this what we get is a fresh water stream of water. That's what we all want. But at the same time there is another stream which comes, which is made up of concentrated saline, which no one wants it, but what we do with it, we normally discharge back in the sea. So there are associated problems with all three components of this process. And the biggest is that it's going to impact on the health of marine health at the interface where these plants will be placed. So the problems are that this process is highly energy intensive, has a big carbon dioxide footprint. Second, at the intake, it takes millions of liters of water into the plant, and with it, it takes many organisms which are in the ocean. Among them are phytoplanktons, fish, larvae, and these are taken, and during pre-treatment, they are killed and removed. And third is environmental impacts we don't know which are associated with by discharging back saline or brine back into the ocean. So what are the solutions? What we are trying to see is we want to study how all these processes will affect marine life. And for this we are investigating or using diatoms, which are a group of phytoplanktons. How does this process affect diatoms? Now these diatoms are very small microscopic organisms. They produce their own food because they can photosynthesize. in numbers, in size, they may be so small, but their contribution to world carbon dioxide fixation or carbon fixation is 20%, which is more than what we get from the tropical rainforest. Then they are very sensitive to any environmental changes in the ocean, and they are taken as or studied as biological indicator for fresh water. They occur in a lot of different species, and at the point or anywhere on ocean, there are communities which are well suited to that environment, and any change in that environment will change that community structures of these diatoms. So what we want to know is what happens to these community structures, or what are the community structures at the point where we are putting these desal plants, and with time how those communities will be changed. Means the sensitive communities which will be affected by any side effects will disappear, and only the resistant will appear there. So that will be indicative that what these processes are doing to this ocean or health of ocean. They are also very interesting organisms in terms of nanotechnology, which I won't touch today, because they make cell walls which are made of nanostructures of silica, and they are considered as the master of nanotechnological engineering. So what we have achieved is that we are now looking at the community profiling of these diatoms at the intake point, as well as where we are outflow is coming of brine, and we are using metagenomics, which is a very next generation sequencing, DNA sequencing technology to which will tell us what different species are there and how, whether they are changed by putting these desal plants at that point. So we are already doing this thing, and then also we want to know that what happens to these organisms when we expose them to brine, which is saline or contains other chemicals, toxic chemicals. And for that we need to get these species isolated from that point and culture them in a laboratory, so that then we can, under controlled environment, we can understand and find out effects of this brine in time. And that's what we have done in last few months. We have isolated a number of species from the point Stanton back, and we are able to culture them, and we are also monitoring their health and how their growth and survival is affected by salinity. And this will give us information that in future what mitigation processes or technology we can then use, so that there are minimal effects of these desal plants on the environment and marine environment. This will create opportunities for collaboration, and which is already done, and also in terms of consultancy, that how to manage our current processes to minimize the effects on nature. And this is a right step in the direction of global water security. And in the end, I would like to thank our funding bodies, which are NCDA and Water SA, Flinders Uni, MISA. Thanks very much.